Hello, my name is Meaghan Rose, and I am a third-year PhD student studying in Dr. Busek Chang's lab. I have been working to isolate and characterize nodulating bacteria isolated from stem nodules of Texas native Aeschynomene indica plants. Now, some plants, like Aeschynomene indica, have the ability to form symbiotic relationships with nitrogen-fixing bacteria. These bacteria fix nitrogen into a usable form for the plant in return for additional carbon sources as well as environmental stability. Now, these relationships are important because crop plants that form these relationships have a decreased dependency on nitrogenous fertilizers, which are not only very costly to produce, but are often inefficiently applied, resulting in runoff and pollution. Now, it had been believed that for this relationship to form, the bacteria must produce certain signal molecules referred to as nod factors. However, it was recently discovered that some bacteria are capable of forming these nodules independently of nod factors. These nod factor independent nodulating bacteria were first isolated from Aeschynomene indica plants native to Senegal, Africa. So, the goal of this work is to isolate and characterize any nodulating bacteria from the stem nodules of Texas native Aeschynomene indica plants in hopes of finding crop inoculants that are better adapted for the Texas environment that can provide not only nitrogen fixation, as well as phosphate solubilization and increased crop yield, as well as increased survivability against extreme abiotic factors. Now, once the plants were located using a series of digital and physical field guides as well as databases, the plants were taken back to the lab, washed, sectioned into small portions, and the nodules were removed using a sterile razor blade. The nodules were then surface sterilized themselves using a series of washes with ethanol and bleach and sterile water. The nodules were then crushed, mixed with liquid media, and then spread out onto solid media so that each of the individual isolates could be obtained and analyzed. Once these isolates were obtained, they underwent a series of tests to determine a few different things. First, their nodulation ability, if present, their dependency on non-factors, if they use them or not, their nitrogen fixation ability, how well they do it, if they do it at all, and their identification is to be determined as well, down to the genus and species level using sequencing. All of this work is done using a combination of growth chamber experiments, acetylene reduction assays, and PCR and sequencing. Very briefly for the growth chamber experiments, the seeds are surface sterilized, placed in their pouch, and inoculated with their particular bacteria. The plants are allowed to grow for a certain amount of time, the nodules are observed, and the bacteria are re-isolated from the plants. Out of all the isolates being tested, one in particular has shown great potential. This is TSN1. TSN1, out of all of them, has shown the most consistent nodulation ability thus far. It does seem to induce nodules on its host plant, Aeschynomene indica. These nodules are typical of what we would expect. They're very small, darkly pigmented. They are located along the taproot, as we see typically in Aeschynomene indica. However, it's also interesting, based on targeted PCR, it seems that this isolate lacks nod factors. So if it is indeed inducing this nodulation, it seems to be doing it independently of nod factors. In addition to this, the 16S sequencing results revealed that this isolate, TSN1, is a particular strain of Bacillus Megaterium. Now, Bacillus Megaterium is very interesting. They are known endophytes of plants. They have been commonly isolated from nodules, and they have been shown to demonstrate or to confer beneficial characteristics to their host plant, such as aid in the resistance of abiotic factors, such as salt tolerance and metal tolerance as well. They've also been shown to help in the reduction of particular diseases, specifically like fungal diseases like Fusarium wilt. And very few of them have actually been shown to induce nodulation themselves. They do, in fact, have nod genes. So the question is, perhaps this TSN1 isolate obtained this nodulation ability from its neighbors. So in conclusion, TSN1 does seem to induce nodulation, seemingly independent of nod factors. This bacteria has great potential as a beneficial crop inoculant, and this work could provide alternative methods to crop maintenance that would be cost efficient and environmentally friendly. Thank you.